Now, scientists in Southampton are working with an innovative model to do initial analysis of treatments for respiratory diseases. The nose in a dish doesn't quite look like an actual nose, but because it's based on human cells, it offers an alternative to animal testing and can give researchers a quick but close insight into infections that enter our bodies through our sensory organ. Our reporter Nicole Ris went to Southampton General Hospital to find out more.
Scientists are getting a closer look at our lung functions when they use the nose in a dish. The model allows them to culture and regrow nose cells in healthy conditions and test drugs.
The nose cells actually mimic the lung cells, so people with lung conditions and people who have sort of deep-seated lung infections, by sampling the nose cells, it actually mimics the lung cells, so it's an easier site to sample. And it's easy access, less invasive for the patient. So we take small cells from the nose using a little brush, and we can culture them on a plastic dish, which allows us to get them to regrow in healthy conditions, and it allows us to investigate whether the cells are working properly, whether the hair-like structures on the surface called cilia are working properly, and also it allows us to test whether if we want to apply drugs onto the cell surface, whether the drugs are not affecting the cells in a detrimental way, harming the cells, whether the cells can respond to the drugs. We can also look at infection and what happens to these cells. So it's basically a model of the nasal cells that encounter the environment.
The nose is where the body first encounters pollution and infections. It's an easy point of nasal drug delivery when it comes to defending against colds, and by imitating the sensitory organ, scientists also try to understand the conditions of a rare disease.
We study a rare disease called primary ciliary dyskinesia, and it's where patients are born with abnormal ciliary function, so the little hairs that should move mucus up and out of the lungs don't work properly, so they're more prone to infection. So what we do here using our high-speed video camera, we record the beating of the cilia, and we use the high-speed video to record at high frame rate, and then we can slow the video down and look to see whether the cilia are beating normally, and this helps us to diagnose their condition. We can also grow their cells in our nose dish culture system, and this allows us to work out whether their condition is a primary condition that they've inherited, or whether it's actually a secondary condition that could be cleared up with treatment.
The model doesn't only benefit further research on diseases, it also prevents animal testing. In the future it'll be applied to other conditions like asthma, and it might also help to develop new drugs to protect against infections.
We're actually looking at the moment to see if we can, what affects nasal spray treatments, and also whether we can invent new treatments to help protect against bacterial infections, such as we have a study at the moment looking at the effects of sugar analogues to see if they can help protect ciliary function during a bacterial infection.
Nicole Ris for That's Solent.